It is my great pleasure to introduce our keynote speaker, Polly Matzinger. She's one of the best immunologists in the world. One of the major things that I can say about Polly is that she's very, very clear in what she says. So she took the challenge of talking in front of an audience of nanotoxicologists, trying to let us understand how reacts to alien factors, including nanoparticles, and how our immune responses decide whether these things are a danger or not. So, Polly, please, the stage is yours.
Thank you. Usually I start by telling people that it's your fault if you fall asleep, so you mustn't fall asleep. Because you fall asleep if I use a word you didn't understand, or I use a word I did understand, but you didn't understand how I used it and you get confused. And so what you need to do is to stop me right then and there and ask me a question. So please raise your hand in the middle of the talk if I lose you. Yeah, I'm going to try not to. So I understand that most of you are not immunologists, right? How many immunologists are in the audience? Oh, quite a few. Okay. Those of you who are, and those of you who are not, but had an immunology class, I'm willing to wager we're taught that the immune system functions by discriminating between self and non-self, and that in fact many of you working with nanoparticles are trying to find ways to make the nanoparticles look not non-self. So the idea is that each individual learns self early in life. Whatever is around when you're in utero or a couple days after birth is called self. Anything that arrives later, like a virus or a transplant, is called non-self. You fight all non-self, you tolerate all self, except under disease conditions like autoimmunity. How many of you were taught that? Okay. I was taught that, and I have to tell you, as a graduate student, I had a terrible, terrible time with this idea, because I had trouble with things like puberty. You know, most of us sitting in this room are not the same as we were in utero or a couple days after birth. And so my professors patted me on the head and said, there, little girl, nothing new appears at puberty. It's just changes of concentrations of things like hormones that you already had. So I pointed out a condition to them that they hadn't thought about, mostly because they had Y chromosomes. And that is this tissue. This is the newly lactating breast. It makes proteins that this body has never made before. And even though most of them go out into the outside world, some of them, like alpha-lactalbumin and beta-lactoglobulin, end up in the bloodstream. And the cells making those proteins have MHC molecules on the surface, which, as you know, are these Schlepper molecules that take bits of the inside of a cell and make a window display on the outside of the cell. And we can presume, although there's no data, that bits of the proteins, milk proteins, are also being expressed on the tissues. So, according to the self-non-self model, the newly lactating breast ought to be rejected because it's making proteins it never made before. And we know that it's not. And when I brought this up to my professors, they said, oh, well, that's because when this individual was learning self, she was drinking mother's milk. And so I pointed out to them that my generation has actually done an experiment. When I was born, it wasn't socially cool to drink mother's milk. You drank Nestle's or Carnation. And although these companies are still trying to do this to third-world women, the important point was that when my generation grew up and started lactating, our breasts didn't fall off. You know, somehow the immune system knows how to deal with a changing self. That was one of the problems I had. Here's another one. It's what Charlie Janeway calls the immunologist's dirty little secret. And that is that you don't respond to everything that's foreign. And people who are trying to do vaccines know that. So, for example, if you want to make a vaccine with an attenuated virus, usually all you have to do is put in the virus. But if you want to make a modern vaccine, a designer vaccine, a protein or a peptide vaccine, people have learned that putting in just the protein, even though it's foreign, doesn't get you an immune response. You have to add something called adjuvant. Right? So for example, if you took a mouse and you injected it with egg white albumin, ovalbumin, definitely foreign to a mouse, it doesn't matter if you inject it intravenously, subcutaneously, intracranially, you don't get an immune response unless you bought it from Sigma and it has LPS in it. You get a response if you add adjuvant. And our favorite adjuvant as experimentalists is something called complete Freund's adjuvant, which is a mixture of mineral oil and mycobacteria. If you add that, you get a nice response. Now that's non-self. The same is true of self. If you inject myelin basic protein into a mouse, from a mouse, nothing happens. If you add CFA, you get a nice response. If you add something really nasty like pertussis toxin, you get an autoimmune, anti-brain demyelinating disease called experimental autoimmune encephalomyelitis that people study because it's kind of like multiple sclerosis. It doesn't seem to matter to the mouse whether what you injected was non-self or self. What matters is you added adjuvant. Now, I had lots more problems when I was a graduate student, but there wasn't a lot of support for asking these questions. There was no support for trying to answer them. And so I spent 10 years postdocing in Cambridge, England, and Switzerland trying to figure out exactly how does the immune system discriminate self from non-self. And then I got a job at the NIH, and there was a postdoc next door named Ephraim Fuchs. And Ephraim was asking the same questions for a different reason. He's an oncologist, and he was asking, why is it that we have tumors that we know express foreign proteins, either because they're virally encoded, or because they're mutated antigens, or because they're proteins that were on very early in development and not at the time the immune system came in. And yet we don't reject them. And by that time I was in old fogey mode, and I patted him on the head and said, there, Ephraim, we all know that tumors are immunosuppressive. And unlike me, he wouldn't let go. So we spent 2 years arguing, and we finally decided that an immune system that fights everything in the air you breathe and everything in the food you eat and the fetuses you carry and the sperm that gets you those fetuses is evolutionarily stupid. A smart immune system would be the one that fights things that are dangerous. That took us 2 years. That's all very well. You know, why should you fight a virus that comes in, makes 3 copies of itself, and leaves? Ephraim says you might want to welcome it because it could bring you genes you couldn't get otherwise. It does you no harm. It took us another year to figure out a definition of danger that works. Now, when I say works, I mean a definition that you can use that explains what the immune system does right and also what it seems to get wrong. And the definition we came up with sounds really simple. I'm amazed it took us a year. And that is that something is dangerous if it does damage. If it doesn't do damage, it's not dangerous. You don't need to respond to it. It took us another year to figure out how does the immune system know that something has entered the body and is doing damage. And the idea there changed our entire view of immunity. And the idea is that the immune system doesn't know. It's told. It's told by the damaged cells. So that what happens is a tissue that gets injured or dies badly or is overheated or overcooled, stressed, sends alarm signals, and that it's those alarm signals which trigger an immune response. Right? So the idea is that if you have a tissue which is healthy and happy and doing its function and expressing on its surface a window display of peptides, of things being made inside, that any lymphocyte, any white blood cell which can recognize that peptide gets what we call signal 1, and that signal 1 by itself tells it to die. So healthy tissues are constantly inducing tolerance to themselves. And if this tissue dies by normal programmed cell death and gets scavenged by either its neighbor or a special scavenger, that's still quiet. But if it gets damaged, it sends alarm signals. And those alarm signals are picked up by resident cells called antigen-presenting cells that live in any tissue that we have studied so far and that are normally asleep. And they can be awakened by alarm signals from damaged tissues because they have receptors for those. And they can also be awakened by bacterial products. And it seems that they can also be awakened by some viral products. So what happens when these cells get awakened? They take in what they see around them. They get rid of the water. They keep the solid bits. They move to a local lymph gland. And there they present on their surface the solid bits of what they have found. So that now, if a lymphocyte recognizes that, it gets signal 1 from having recognized it, and it gets a second signal that we call co-stimulation, and that second signal is what activates an immune response. If you don't activate an antigen-presenting cell, you don't get a primary immune response, period. And what the danger model says is that what activates them are mostly alarm signals from damaged tissues. We can come back to the bacteria. It turns out that if you make that one assumption, that alarm signals from damaged cells activate the immune system, and healthy cells don't, you can explain almost everything the immune system gets right, and almost everything it seems to get wrong. So here's the list. We don't have time to go through it. I'm going to take some examples. Here's the list of things you can explain. You can explain why you don't kill yourself at puberty, or at least not immunologically. You can explain why lactation is not a problem. You can explain why mothers don't reject their fetuses, and we will come back to that. You can explain why surgical transplants, which carry foreign antigens, are usually rejected, but tumors, which also carry foreign antigens, are not. You can explain why you get graft versus host disease with a bone marrow transplant. You can explain why there are parasites like filaria, this is the worm that gives you elephantiasis, that live in intimate contact with the immune system in the lymphatics, and yet you don't usually get an immune response. And at least for the first 15 years of this model, I couldn't explain allergy and asthma. And it didn't bother me. And the reason it doesn't bother me is that the danger model was designed to answer one question the immune system has to ask when faced with something. And I think allergy and asthma is a problem in the second question the immune system has to ask when faced with something. So the first question is, do I respond or not? The self-non-self model said you respond if what you're looking at is foreign. Charlie Janeway had a model that said you respond if what you're looking at is a bacterium. The danger model says you respond if what you're looking at is causing damage. Once you decide to respond, you have a second question to deal with. What kind of response am I going to make? How do I know to make a killer to catch a virus or IgE to catch a worm? The problem with allergy and asthma is a problem in this second question. There are two questions about allergy that people ask. The first one is, what makes an allergen an allergen? And this is important to you as an audience, whereas I think the second question is not. What makes an allergen an allergen? Why is it that the house dust mite has at least 22,000 proteins and 2 of them are allergens? Cats have a lot of proteins and 1 of them is an allergen. That's the first question. That fits under the danger model. I predict that an allergen is an allergen because either it does damage, or it's packaged with something like bee venom that does damage, or it mimics an endogenous alarm signal. We know that DERP1, which is the main allergen from house dust mite, is a protease that attacks the surface of lung epithelium and also of B cells in the immune system. So what makes an allergen is that it's immunogenic because it causes damage or mimics an alarm signal. Why some people respond is the second question. And I think that that's the wrong question. I think we all respond to allergens. It's just some people make IgE, which is a problem, and some people make IgG, which is not a problem. So I've been suggesting to allergists for about 10 years that they should look at the non-allergic siblings of allergic kids and ask if they're making immune responses to the same allergens. And I would bet a case of champagne that they are. They're just not making IgE. They're making a normal IgG response. And so what we need to ask there is what controls the kind of immunity we make. Right? We don't have time to go into that today, but I just published about a month ago a theory about that. So let's take 3 transplants, 3 tissues that have foreignness about them, and ask why is one of them rejected and the other 2 not. Right? The self-non-self model would predict that fetuses should be rejected, transplants should be rejected, and tumors, because they have foreign or mutated proteins, should also be rejected. Charlie Janeway's model suggests that fetuses should not, because they're not covered in bacteria. Transplants should not. You know, most well-done heart transplants are not covered in bacteria. And tumors should not. Right? What does the danger model predict? So let's start with mothers and fetuses. Peter Medawar, who was one of the 2 people who shared the Nobel Prize for the self-non-self model, already knew in 1953 that fetuses were a real problem for the model. They're foreign, they ought to be rejected. And in his inimitably arrogant, Oxford-educated Britishness, it never occurred to him that the model might be wrong. He suggested instead that there was something wrong with female physiology. He said, fetuses immunosuppress their mothers. Now, that's evolutionarily really smart. You take the individuals that are carrying the next generation and turn them into AIDS patients. It's not smart, and it turns out it's not true. Pregnant individuals, actually, how many of you have carried fetuses? Did you have to live in a bubble? No! Has anybody noticed? You know, all those sheep out there on the hills, they don't have to live in bubbles. Medawar had kids. I don't know where he was when his wife was pregnant, but she wasn't in a bubble. Pregnant mice reject skin transplants from the donors of their fetuses perfectly fine. Now, it's true that there are some parasites, like malaria and toxoplasma, that take advantage of the pregnant state. But it turns out it's because the parasites are smart, not because the immune system is not on. In general, pregnant individuals are pretty healthy. Right? So, not evolutionarily smart idea and not true. So Medawar actually suggested a second one, and that is, by the way, also still in textbooks, and that is that the placenta is both a physical and immunological barrier between mother and fetus. So that a few antibodies get across, but in general, the immune system can't get in there to see the fetus. Now, that's also pretty smart. It means that if your fetus gets infected, you can't do anything about it and you're dead. And it's also not true. There's traffic in both directions across the placenta. There are people working on isolating fetal cells that are in the maternal circulation as a way of getting tissue to do a non-invasive prenatal diagnosis. So that says that there's traffic from fetus to mother. There's also traffic from mother to fetus. That was first discovered at Eurotransplant by Jan van Root's group in Leiden, where they have people waiting for kidneys. And if you have people waiting for a kidney donor and you have a living donor offering their kidney, you always test the immune response of the recipient first before you do the transplant. Because if there's already a strong response, you don't want to waste the kidney. So they test all their recipients against the donors. Now, mothers are often donors. And it turns out that when they looked at the immune responses of their kidney potential recipients, 25% of them did not make an immune response to their mothers. They responded to the non-inherited transplantation antigens of their fathers, but not the non-inherited transplantation antigens of their mothers. And Lee Zhang and Rick Miller followed this up in Toronto, where they could use genetically marked mice to tell the father, the mother, and the F1 progeny, and they found that 25% of adult mice are microchimeric. They're carrying a small number of maternal hematopoietic cells. 50% of human kids, these are kids who do have to live in a bubble, have T cells from their mothers. So that says that there's traffic from fetus to mother, and there's traffic from mother to fetus, and yet the fetus is not rejected. Why not? Because it doesn't look immunologically dangerous. It doesn't matter that it's foreign. The kind of death that goes on in fetal life is programmed cell death. You know, you want to make a hand out of the primitive paw, you kill the cells in between. That's apoptosis. It's programmed cell death. The cells get scavenged by their neighbors. There are no alarm signals. There's no immune response. And should the fetus get infected, there will be alarm signals. You will get an immune response. And if in the process of clearing the infection, you also clear the fetus, well, at least you're alive to have another one. Right? And the medical literature tells us that spontaneous miscarriage, spontaneous abortion, is a common concomitant of infection during pregnancy. So when I teach the danger model to grammar school kids, I give them this little story. The old model, the self-non-self model, looks like this. If you think of the body as a community, the immune system are the police. Right? They're constantly going around looking for foreigners. And if they find a foreigner, they shoot them. And they define a foreigner as anybody they hadn't already met when they finished grammar school. Right? The new model says, no, no, no. If you think of the body as a community, the immune system are more like firemen. They sit in their fire stations playing cards until somebody rings an alarm. And it doesn't matter if the person ringing the alarm is a traveling salesman or tourist, an immigrant, or somebody who grew up there. And it doesn't matter if the fire was set by any one of those groups of people. The firemen come out when they hear an alarm. And unlike the cops, they have different responses. They bring a different truck if it's a cat up a tree than they do if it's a 3-alarm fire. Right? So the danger model says the immune system is more like firemen. You don't respond to your fetus because it isn't ringing alarms. So if that's the case, why do we respond to a surgical transplant? Where is the danger in a transplant? Anybody? Sorry? Surgery. Absolutely. Surgeons are dangerous. Yeah? They cut, they burn small vessels, they put organs on helicopters and fly them around the country. They get ischemia and post-reperfusion ischemia. This is not programmed cell death. The prediction is, if you could do a transplant without doing damage, if you can get something into the body without doing damage, it shouldn't be rejected. Now, you can't do a transplant without doing damage. Even a blood transfusion. You take the blood, you cool it. That's called a heat shock. You wake up the antigen-presenting cells in the blood. By the way, when we grow them in culture, you look at them cross-eyed and they wake up. We grow these cells from bone marrow, and we grow them for 6 days. And if day 6 happens to be a Friday, we have more spontaneously activated cells than if day 6 happens to be a Monday. And it's because the incubator doesn't get opened as much during the weekend. Just those changes in gas and temperature is enough to spontaneously activate some of these antigen-presenting cells. Okay? So you can't do a transplant without doing damage. However, you can do an experiment. And Colin Edison, when he was in my lab, did this experiment. He now has his own. He's doing it again. So I'm showing you the preliminary data. He took skid mice. So these are mice with no adaptive immune system. They are the mice who have to live in a bubble. And under the kidney capsule, he gave them a heart transplant of a neonatal heart that differed by enough transplantation antigens that if this mouse had had an immune system, it would have rejected it in anywhere from 14 to 26 days. But it doesn't have an adaptive immune system, so it can't reject the heart. So he left the heart to heal for 9 months. And we asked, now with the heart healed, with it not sending alarm signals, would it get rejected if he gave this mouse back an immune system? So he did. And he waited. And the hearts were not rejected. If he had given them a heart and only waited 1 month before giving them an immune system, they're rejected. 1 month is not enough time to heal. 9 months, they're not rejected. So he then asked the second question. If you could leave the heart and the immune system to interact with each other, would the heart induce tolerance of itself? Remember I said that the lymphocyte, if it sees that antigen in the absence of danger, will become tolerant. So he tested that by leaving the heart and the immune system to interact for 5 months. After 5 months, he immunized the mouse with cells from the heart donor and looked for killers, which is one way to assay the immune system. Now, people say I never show data. Here's some data. What you see here are 3 groups of mice. The control mice sat in the same cages. They were litter mates, so they sat in the cage for 9 months. They didn't get a heart. After 9 months, they got an immune system. They sat for another 5, and then they were immunized. And we tested them for killing by titrating in the killer cells. And you see that as you add killers and look for kill, 2 of them are killing phenomenally well. 1 of them is killing really well and not as good as the other 2. And it's donor-specific because it doesn't kill self. These are the 3 mice that had had their hearts for 1 month before they got the immune system. They rejected the hearts. They didn't even have to be immunized, and they make nice killers. And these are the 3 mice that had had their hearts for 9 months. And you see that not only did they not reject, you couldn't immunize them to reject. They had become tolerant. Now, that says that if you can get something into the body without doing damage, or if you can block the immune system long enough for it to heal, you will not reject it. Now, when I gave these data to the annual meeting of the American Transplant Surgeons Society, the surgeon got up in the back of the room and he said, "You're wrong, dearie." He says, "I have kidney patients who have had their kidneys for 15 years, and some of them get sick of the side effects of cyclosporin and go off their meds. And depending on what center they're in, 50 to 90% of them now reject their kidney. So 15 years ought to be long enough for a kidney to induce tolerance to itself. So luckily, it turns out that what's wrong here is not the model, but the drug. Remember, I said that the way the body induces tolerance is that a tissue is happy and healthy, and it's expressing, as a window display, its self-peptides. And if a lymphocyte sees it, it gets signal 1 from that recognition without co-stimulation and dies. Now that means that in order to induce tolerance, the lymphocyte has to get signal 1. It has to recognize self or non-self and get signal 1. Cyclosporin blocks signal 1. It blocks the calcium flux, which is the result of the lymphocyte's ability to recognize them, or the act of recognizing it. So effectively, what you're doing with cyclosporin is you're blindfolding them. And for 15 years, they're wandering around. blindfolded. And then you take it off. And they're fine until they get infected. So for a while I was getting emails from a woman who was a heart transplant recipient who got sick of the side effects of her meds and went off them. Now that's a brave thing to do, because if you lose your kidney, you're back on dialysis. If you lose your heart, you're dead. She was fine for 18 months. And then she got a recrudescence of an Epstein-Barr virus infection and started to reject her heart. And so they put her back on her meds. But what this says is, if we could block signal 2, co-stimulation, if we could mimic the body's own way of inducing tolerance, we ought to be able to get transplants to take. And there's one guy who tried that. His name is Kirk Alyn. Actually, believe it or not, Commander Kirk. He's in the Navy. He's a transplant surgeon. Soldiers often need transplants. And he did an experiment in monkeys that was published about 5 years ago in Nature Medicine, where instead of blocking signal 1, which is what cyclosporin does, he blocked signal 2. He mimicked the body's own way of inducing tolerance. And he gave kidneys to monkeys. And by the time he published this, one monkey was about 5 years out. Yeah? What he did is he blocked signal 2 for 5 months and then he stopped. 6 monkeys, none of them rejected. It just shows you, if you have to get down to the details. God is in the details. You got to know which signal to block. You want to mimic the body's own way of doing it. He started, by the way, this is kind of sad. He started a clinical trial. He blocked one signal 2. There are actually 3 of them. There are probably 4 of them now that we know. He wanted to block all of them in humans, and he couldn't. And the reason he couldn't is that one antibody was owned by Chiron, another one was owned by Immunex, and a third one was owned by yet another drug company. And the drug companies wouldn't come together to take responsibility for the same trial. So Kirk's now making his own. And of course, it's taking him a while because he's got to make his own in a way that gets around the patents. Yeah. It's a problem. Yeah? We have these things that work, and yet the companies won't let us try them. Okay, so you reject a transplant because it looks dangerous. You don't reject the fetus because it doesn't look dangerous. Why don't we reject a tumor? What's wrong with a tumor? It's too healthy, right? It's lost growth control. So the whole time that the tumor is growing, especially early on, it's multiplying or dividing. It's not sending alarm signals. And so the immune system can't discriminate between rapid cell division in the bone marrow, rapid cell division in the thymus, rapid cell division in the gut and the skin, and the rapid cell division of a tumor. No alarm signals, no immune response. And what's happening is, if this tumor is expressing some tumor antigen while it's growing, and if a lymphocyte can see that, it's going to get signal 1 without 2 and die. So by the time that tumor is big enough that if it's a solid tumor and poorly vascularized, it starts to starve in the middle and die, most of the cells that can see it have been tolerized. And by the way, starvation death is programmed cell death. And so that doesn't send alarm signals either. You'll start getting alarm signals when there's so much death in the center of a tumor that you've overloaded the local scavenging capacity. And then you might get an immune response. Right? And then the 3 T cells that are left that can see that tumor will get turned on, because the presenting cells will pick up tumor antigens, take them to the local lymph glands, present them to the T cells that are passing by, and they'll get turned on. But what happens then is each lymphocyte, it is estimated, can kill 3 to 30 targets before it needs to be reactivated. So you've got 3 left. They become about 300. Each one of those 300 can kill 3 to 30 tumor cells. After it's killed its 3 to 30 tumor cells, it drains back to the draining lymph gland. It needs to be re-stimulated. That takes another 3 days. And then they go out and kill another 3 to 30 tumor cells. In the meantime, what's the tumor doing? It's making millions. You've lost the race. So Steve Rosenberg, who's America's Mr. Cancer, actually had a brilliant idea. He said, let's get those few cells that are in the tumor and expand them and put them back in. They're called tumor-infiltrating lymphocytes, or TILs. So he got some tumors, he took out the TILs, he expanded them in bathtubs full of IL-2, which is a growth hormone for these cells, and put them back in. And if you heard him about 10 years ago talk, he would have a pair of slides he loved to show of a guy who had a melanoma metastasis in the lung, the size of a softball. And he gave him TILs from his own tumors, and it went down to the size of a baseball. And then he did it again, and it went down to the size of a golf ball. And then he stopped, and the tumor grew out and killed the patient. Now, what's wrong here? What's wrong is that he's in what I call vaccine mode. And most people who do immunotherapy for tumors are in that. What is that? It's that you prime and you boost and you stop. Those of you who want to use nanoparticles for vaccines need to listen to this. It works for viruses. You prime the immune system, you expand the number of cells that could see that virus, you boost the immune system, you expand them a little more. That's about all you're going to get expansion-wise. And then they go into what's called a resting memory state. And when the virus comes back, it does the damage that reinitiates that response. But killers kill by inducing programmed cell death. The immune system doesn't throw fuel on the fire. So when you kill a tumor cell, it doesn't reinitiate the response. You need to keep doing it. Right? The resting memory state doesn't do you any good. So I've been telling Steve Rosenberg for 15 years now that he needs to keep boosting. And he won't do it. But there's a guy in Pamplona who has done it, and he published in the Journal of the National Cancer Institute about 2 years ago a paper where he took 20 people who had relapsed of a myeloma, which is a kind of immune tumor, and he made a vaccine for each individual person, and he just kept boosting them. And even though these people had already relapsed once, they didn't relapse again. One guy died. And I wrote to him and I said, what happened? And he said, we ran out of vaccine at 2 years, and he died at 2.5. So what that says is, if you want to keep an immune response going, you have to keep boosting. Okay? So, you don't reject a fetus because it doesn't look dangerous. You reject a transplant because it does look dangerous. You don't reject a tumor because it doesn't look dangerous unless you make it look dangerous. And by the way, I train sheepdogs. I train border collies. I was here actually in Tullamore in 2005 with the U.S. team for the World Sheepdog Finals. It was fun. Yeah? I was so amazed that we had a country that didn't hate Americans we had Bush then. You guys didn't hate us. Anyway, I train sheepdogs. I'm out in the sun a lot. I get these little basophiliomas on my nose. Tumors. I'm also sensitive to nickel, so I get some pair of cheap earrings. I break off the part that goes through the ear. I take the flat part, which almost always has nickel in it. I stick it to my nose. I get a great immune response. The tumor goes away. I keep sticking it there until it's gone. Yeah? I don't have to go to doctors. Okay. If you want an immune response, you have to keep boosting. If you want to use nanoparticles as a vaccine, you either want to get a nanoparticle that's going to deposit itself and keep doing damage, or you're going to have to keep giving it. If you want a nanoparticle that doesn't give you an immune response, you need a nanoparticle that does no damage. Okay? Now, not only that, it should do no damage, and it should not mimic an endogenous alarm signal. So what are the alarm signals? Well, the list is getting longer. DNA, the CPG sequences in DNA. There's a lot of them in bacteria. There's also a lot of them in us in the promoter regions of open genes. RNA, or its mimic, poly-IC. Heat shock proteins. So 60 is a little controversial, but 70 is now known to be an alarm signal. GP96 is a heat shock protein, a chaperone that's known to be an alarm signal. Hyaluronic acid, the breakdown products. Hyaluron lines your blood vessels. It's a polymer. And the breakdown products of hyaluron that you get, for example, when you're bruised or cut, act as alarm signals. ATP is an alarm signal. Cardiolipin, which is a lipid from the inner membrane of mitochondria, is an alarm signal. LPS, lipoteichoic acid, lipoteichoic acid. In general, things that have what my postdoc song calls hippos. So a hippo is a hydrophobic portion. Hydrophobicity by itself is an alarm signal. In fact, it's an alarm signal that goes back to single-cell evolution. Because it's kind of interesting, the property of hydrophobicity biologically is both useful and dangerous, right? Very early in evolution, it was useful because you can make membranes with it and keep the outside out, the inside in. You can use it to glom proteins together, in the inside and hold their structure. But it's dangerous because hydrophobic things kind of non-specifically aggregate. And that's not good for a cell. And so cells go to great lengths not to allow hydrophobic portions to be exposed. Right? For example, when you've got message and you're making protein off the message and you've got the hydrophobic parts of that protein exposed, they're covered by chaperones. There's a lot of lipid handling molecules inside cells. There's also a lot of lipid handling molecules in blood. There's a protein called LPS binding protein. It's misnamed. Its real function is to shuttle membrane lipids from cell membranes to HDL and back. There are proteins that are involved in dealing with dietary lipids. The upshot of all that is that lipids are not normally exposed. The hydrophobic portions are not normally exposed. And very early in evolution, cells learned to see that if they were exposed, it was a sure sign that something bad had happened. And so we got systems like the inflammasome, which can see perturbations in membranes, the exposed hippos, which turn on repair and remodeling systems. And my postdoc song got this idea that perhaps the immune system piggybacked on that. And what we know now is that the receptors on the outside of presenting cells that are used to activate them and start an immune response mostly look at things that are hydrophobic. The receptors on the insides of cells, also some of them, inflammasome, for example. But there are others that look at DNA and RNA because DNA shouldn't be in the cytoplasm. So if you've got DNA in the cytoplasm, something's wrong. So Song suggests if you want to get something into a body and not get an immune response to it, make it hydrophilic, not hydrophobic. You probably already do that because hydrophobic things aggregate. Right? These are some of the alarm signals. Uric acid, crystals, is an alarm signal. This is the list that we know so far. People are working on this. There are more and more of them coming out every day. I have suggested that anything which is normally inside a cell and not outside a cell can act as an alarm signal. My favorite is mitochondria. Why? Because they bring two of the main immunological models that people have been arguing about for about 15 years together. Charlie Janeway suggested that the immune system responds to bacteria. I'm suggesting they respond to things released by damaged tissue. The mitochondrion, of course, is both. And it turns out that if you inject mitochondria into rats, you can mimic something called systemic immune response syndrome, which is what people with major trauma die of. There's a trauma surgeon at Harvard named Carl Hauser who published a paper in Nature about a year ago looking at a phenomenon that nobody understood. So 50% of people who get major trauma, crushed by an elephant, hit by a car, really bad, they die. You can't predict which ones are going to die, but 50% of them approximately die, and they die of what looks like sepsis. They get a cytokine storm, they get multi-organ failure. It looks like a massive bacterial infection, but nobody can find any bacteria. And what he showed was it's the mitochondria released by all that damaged tissue that turns on neutrophils. And it's because neutrophils have a receptor for formulated methionine peptides. So bacteria, when they start making a protein, the methionine that they use is formulated. We, when we start making a protein, don't. But mitochondrial peptides are also formulated, and neutrophils have a receptor for that. So they can recognize both bacteria and mitochondria. So if you've got something that affects your mitochondria, you might want to use it for a vaccine and not for something where you hope not to get an immune response. So, if you don't want to get an immune response, don't do damage. Don't mimic an endogenous alarm signal. Don't mimic an exogenous alarm signal. And how do you know? There are tests you can do. So, one of them is to grow some of those presenting cells. Grow dendritic cells. Don't disturb them. Don't look at them cross-eyed. Don't change the temperature of the medium, and just drop your stuff in the well and see if they get activated. There are lots of ways of measuring activation, and you should measure lots of them because they can get activated in one direction or another. Now, that won't always do. For example, people studying hemophilia, they're injecting factor VIII into kids that are bleeding, have wondered why it is that these kids start making antibodies. Turns out that, of course, they're giving it to them at the time when they have activated immune systems because they're bleeding, right? Dangerous times. When they tested factor VIII, recombinant factor VIII, to see if it would activate presenting cells, by itself it doesn't. But when they give it to somebody who's bleeding, they eventually start making antibodies. So there's a trial going on. There was a preliminary trial that if they gave it to the kids when they were not bleeding, would they make antibodies? And the answer was no. Alum is the most commonly used adjuvant in humans. If you drop that on dendritic cells, it doesn't activate them. What it does is you stick it into a muscle, and it does so much damage in that muscle that it activates the local presenting cells. In fact, alum is 1,000 times more effective if you use it subcutaneously than if you use it intramuscularly. So why do they use it intramuscularly? Because no parent would bring their child back for a second vaccine if they saw the damage that was done by the first one. So they stick it deep into a muscle so you can't see it. But it means that sometimes if you want to know if your product is going to cause an immune response, just dropping it on the dendritic cell is not enough. Stick it into a muscle. Put it under the skin. If you put something under the skin of a mouse and it's innocuous, and we've done that, for example, with apoptotically dying cells, cells that we drove to apoptosis using ceramide, it doesn't give you an immune response. If you kill them by freeze-thawing them so that their innards come out and put it under the skin, now that gives you an immune response. So there are ways of testing. So that's it. I hope that what I've convinced you of is that the foreignness of your product is not necessarily what's important. It's the dangerousness or the mimic of dangerousness that's important. Thank you for listening. Any questions?
Does anybody dare to ask a question?
Oh, come on. Can I wager there's at least one person in the audience wondering about Rh disease? Why do mothers make responses to their Rh positive babies if they're Rh negative? Anybody? You were wondering? So it's important to bring this one up because it shows that God is in the details. The details really matter. The important thing about Rh disease, so you have an Rh negative mother, an Rh positive father, the fetus is Rh positive. it's not a problem in the first pregnancy. Right? Pregnancy is not the problem. It's the problem in the second. Women get primed at birth. Birth is dangerous, right? Ripping, bleeding, surgeons with knives, dangerous period. And in fact, physicians know that, because what they do is they give an antibody to block the Rh factor at the time of birth so that the mother's immune system doesn't get primed. You have that, yeah. That's why you were wondering, yeah? So it's okay, we know about it. But what it's telling us is that the mere presence of that foreign molecule all during pregnancy is not the problem. It's when it becomes associated with damage that it's a problem. Wow, I've convinced everybody.
It seems so. I think that maybe we can break for coffee. If you want to ask questions directly to her, she will be around. I have a little present for you.